Every cell in our body has a genetic code that determines its structure and function. Just as a computer stores data in the form of zeros and ones, the cells in our body store genetic instructions in the form of 4 chemical bases: adenine, thymine, guanine, and cytosine. These chemical bases are found in our DNA arranged in a specific order. This order or sequence is what determines biological traits. The sequence must be replicated so that it can be transmitted during cell division and ensure that all cells have a copy of the genetic code to dictate their structure and function. However, DNA has a complex double-helix structure, so how exactly does it replicate? In the 1950s, scientists proposed 3 popular models to explain how DNA replicates: the semi-conservative model, the dispersive model, and the conservative model. In a semi-conservative model, the 2 strands of the double helix separate and serve as a template for the addition of complementary bases in the correct order to produce the daughter strand. As a result, each newly formed DNA molecule will have one parental strand and one daughter strand. The original strand is thus conserved, but it is now bound to a new daughter strand. As a result, this model is known as the semi-conservative model. In the dispersive model, the parental strands are fragmented and the broken ends are joined with newly formed strands. The daughter strand is thus dispersed in the new DNA molecule in this manner. Finally, in the conservative mode of replication, the DNA molecule does not unwind to serve as a template. Instead, the nitrogenous bases rotate slightly away from the interior of the helix. This allows the 2 strands to be used as a template to create 2 daughter strands. When the replication is finished, the nitrogenous bases rotate back into place and the original strand separates from the daughter strand. The original DNA molecule is thus completely conserved. As a result, this model is referred to as the conservative model. These 3 theories were put to the test by Matthew Meselson and Franklin Stahl in 1958. They accomplished this by utilizing 2 nitrogen isotopes. Nitrogen is an essential chemical constituent of DNA. As a result, every time DNA is replicated, new nitrogen atoms are added to the newly formed DNA molecule. Nitrogen has 2 stable isotopes, nitrogen 14 and nitrogen 15. The chemical properties of these isotopes are similar, but their masses differ. Nitrogen 15 is a rare type of nitrogen that contains an extra neutron. Because of the extra neutron, nitrogen 15 is a heavier isotope. As a result, DNA molecules containing nitrogen 15 are denser than those containing nitrogen 14. Nitrogen 14, the lighter isotope, is typically found in DNA. However, Meselson and Stahl grew E. coli in a culture that contained N15 as the only supply of nitrogen for several generations in order to incorporate the heavy nitrogen into the DNA of E. coli for their experiment. As a result, N14 was replaced by N15 in the bacterial DNA. After obtaining a sample from this, they grew the bacteria in new media that only contained the lighter N14 in order for the succeeding generations of the bacteria to incorporate N14 into their DNA. After each round of replication, which takes about 20 minutes, samples were taken from this bacterial culture for analyzing. These samples contain bacterial cells that were produced after the first, second, and third round of replication. Meselson and Stahl utilized cesium chloride density gradient centrifugation to examine the density of DNA in these cells. Cells were lysed for this method, and the resulting crude extract was then added to a solution of cesium chloride. Cesium chloride is a heavy salt. Because the salt is much denser than the DNA molecules, DNA floats on top of the solution. When this solution is rapidly spun in an ultracentrifuge for many hours, the high centrifugal force tries to push the dense cesium ions towards the bottom of the tube. As a result, the concentration of cesium ions is greater near the bottom of the tube than at its top. Consequently, a density gradient is created. Because the solution is less dense at the top, the DNA molecules can now migrate through it until they reach a point where the solution is too dense for them to advance any further. Because N15 DNA is denser than N14 DNA, it can travel further towards the bottom of the solution. Once the DNA molecules have been separated, they can be detected using UV light. DNA molecules will absorb ultraviolet rays, thus casting a shadow on the photographic film placed beneath the tube. The photographic film will now have bands that represent DNA with varying densities. This was done for all the samples that were collected. Meselson and Stahl noted that the heavy band had disappeared after the initial round of replication. Instead, there was a band with an intermediate density. Following the second round of replication, a lighter band had emerged. It's interesting to note that after the third replication, the lighter band's intensity increased. These band patterns matched exactly what was predicted for the semi-conservative replication. The heavy band contains the N15 DNA. Following the first round of replication, the newly created molecules would have one N15-based parental strand and one N14-based daughter strand. This hybrid molecule would have an intermediate density. After repeated rounds of replication, the amount of hybrid DNA will remain constant, while the amount of N14 DNA will continue to increase. Meselson and Stahl's experiment also disproved the other replication models. See, if DNA replicated using the conservative model, then after the first round of replication, there would have been a heavy DNA band and a light DNA band. And if DNA replicated in the dispersive mode, there would have been only one band after each round of replication, with the density of the band decreasing with each round. Hence, with this experiment, Meselson and Stahl demonstrated that DNA replication is indeed semi-conservative. Now, there were still many questions to be answered, like how does DNA unwind and how are the nucleotides added to the daughter strands? I'll be discussing this in detail in my next video, so stay tuned. Thanks for staying till the end. If you like this video, do hit the like button and don't forget to subscribe for more videos like this.